Hi friends, today I wanted to discuss about this skill-based questions in biology, especially diagram-oriented questions. So you have to draw a neat label diagram with labels. Generally, in most of the question papers, you can see that the diagram-oriented questions are more than 10 to 12 marks. There has been instances of question papers having 21 marks diagram based questions too, so such instances are also there. So diagram based questions are quite easy to answer. Only thing that you have to remember is in most of the cases the diagram does not have marks, but correct labeling fetches you the marks. So half mark would be there for labeling. The questions can be from 2 mark questions or 5 mark questions. So if it is 2 mark questions, minimum 4 labelings has to be done, correct labeling with diagram. Then if it is 5 mark questions, it has to be 10 labelings with half mark each for the labeling and a neat diagram. So do not spend too much of time drawing the diagrams, but make a rough sketch of it and label it properly and see to it that the technical details of the diagrams are maintained. Even though there are no marks for diagrams, but at least the diagram should look like what you are supposed to draw. So let us proceed with this diagram based questions. The first is, this is very rarely asked, a diagrammatic representation of LS of flower, but in case if they ask you, you have to draw this flower structure where you have a ovary which is inferior ovary enclosed by the receptacle or thalamus. This is the sepal and this is the ovary. You can write about the parts of the stamen, that is filament, anther and connective, and the style of the ovary which ends up with stigma. Also label the petals. So these labelings, it can be given for 3 mark questions since they have nearly 7 labelings. So 3 mark questions you can expect such diagrams. This is for 1 mark question or question with description of the pollen grain. They have a vegetative cell and generative cell. You can also label the outer exine and inner intine. If they have asked for typical diagram of a pollen grain, for 2 marks you can draw this diagram. So this has been asked for 3 mark questions as well as for 5 mark questions. 5 mark they might also ask you the description. So the transverse section of microsporangium, so you have to draw the ground plan of the diagram with all the labelings like connective, epidermis, endothelium, sporogenous tissues, tapetum and middle layers. Then there is an enlarged portion of this layers where you are going to write the cellular details of epidermis, a single layered endothelium, 2 layers of middle layers, tapetum and inner microspore mother cells or sporogenous tissue. If they have asked you young microsporangium or young anther wall, you can write this. But if they have asked you the mature anther wall, so instead of drawing this microspore mother cells, you can draw the pollen grains. They have asked description of embryo sac with diagram. So nearly 3 marks for the diagram and 2 marks for the notes part it has. So a mature embryo sac towards the micropylar end, it has the egg apparatus with 2 synergids and an egg cell. The egg apparatus consists of 2 synergids and an egg cell, and you should remember to write the filiform apparatus in the synergids. Then there are 2 polar nuclei within the central cell, and towards the chelazal ends there are 3 antipodal cells. So totally it is 7 celled and 8 nucleate mature embryo sac that you have to remember about. So the development of embryo sac is shown here. It has been rarely asked during examination, but in case if they ask it, you should be remembering about that. So the microspore, megaspore mother cell undergoes meiosis 1 to form 2 nuclei and meiosis 2 followed by mitosis, and there are 4 nucleus at each stages. So 3 of them are going to get organized at the micropylar end to form the egg apparatus with 2 synergids and an egg, and one of the nuclei moves towards the central cell and they are forming 2 polar nuclei. 3 nucleus move towards the chelazal region and they form the antipodals. Polar nuclei later fuse to form the central nucleus or secondary nucleus or primary. After fertilization, it's called as primary endosperm nucleus. So the structure of embryo, development of embryo. So the fertilized embryo sac showing zygote and primary endosperm nucleus. After fertilization in the embryo sac, the synergids degenerate and the egg cell, which is fused with one of the sperm cells from the pollen grain, they forms the zygote, and another sperm cell which fuses with the polar nuclei, they are going to form primary endosperm nucleus and they are triploid. So this is the primary endosperm cell. So there is degeneration of antipodal cells. So this develops into the zygote further develops to form the embryo. They undergo the arch-shaped embryo with suspensor, and the mature embryo has plumule with 2 cotyledons and radicle is there, and suspensor is present but it is reduced in structure. The typical dicot embryo, it has plumule with 2 cotyledons, hypocotyle, radicle and root cap, and this is a monocot embryo where it has scutellum. Scutellum is the cotyledon, we call it as scutellum. There is coleoptile which is going to protect the shoot apex, and there is epiblast, and the radicle, it is produced by coleorhiza, and root cap. The coleorhiza protects the radicle. So this is the typical monocot embryo that you are expected to draw. If they are having 6 labelings, they are 3 marks questions, you can expect them. So the male reproductive system, generally you are going to draw this diagrammatic view of male reproductive system. The part of testis is open to show inner details. This is the diagram that frequently it is drawn. Even the other diagram you can draw that, but whenever they have specified part of testis open, you have to draw this. So here you can notice the glans penis which is covered by a foreskin, and all this labeling you have to do: testis, urethra, bulbourethral gland, prostate gland, seminal vesicles. Then there is urinary bladder, ureter, vas deferens, and the open part of the testis you can see the epididymis, vasa efferentia, reti testis, testicular lobules. So these are all the labelings that you have to draw. Majority of the time this diagram is what you have to draw, and you are going to get 5 mark questions for just for the diagram with 10 labelings, half mark for each labeling. Then the next one is, this might be given for 3 marks, the diagrammatic sectional view of seminiferous tubule. So there is spermatogonium, Sertoli cells, primary spermatocytes, secondary spermatocytes, spermatids, which are going to function later as the sperms. So the spermatozoa. So all this labeling of seminiferous tubule, for 3 marks they might give you, where you have going to get 6 labelings with half mark each. The typical structure of sperm cell has been asked for 5 mark questions. So you can have 10 labelings here: structure of head, plasma membrane, acrosome, nucleus containing chromosomal material, neck region, middle piece, mitochondria, so tails. So structure of a human sperm, this has been asked many times for 5 marks or for 3 marks. So you have to remember to have all the labelings there, and sometimes they might also ask you for 3 marks the diagram and 2 marks for description part of it. So this also has been frequently asked question, 5 marks frequently asked diagram oriented question, the sectional view of the female reproductive system. So you have to draw from the tubular region of the vagina, which leads into the cervical canal within the cervix. So the uterine wall has outer perimetrium, middle myometrium and inner endometrium. So they also have a uterine fundus and the uterus or uterine cavity. So the fallopian tube consists of isthmus, ampulla and infundibulum. They have finger-like protrusions called as fimbriae, and they have ovary. So the isthmus, ampulla and infundibulum together, they are called as fallopian tube. So all this 10 labelings carries half mark each. So labeling is very important, and appropriate labeling you have to do. So this is frequently asked question which you should practice for your examination. The next diagram has been asked for 3 marks. I think 3 marks for with description in the previous examination, that is March 2019. So you have to draw the diagram and mention the various parts of the mammary gland, the fat region, the mammary lobe, mammary alveolus, mammary duct, ampulla, lactiferous duct, and these many of the lactiferous ducts, they are all going to fuse to form the nipple, areola. So, and the muscles that are there, the pectoral majoris muscles between the ribs and rib. All this labeling, it could be fetching you around for 5 marks also they can ask this question, or last year they had asked it for 3 mark question with description. So just practice this diagram to once or twice. It has been asked in the examinations, to board examinations too. The next is DNA double helical structure of DNA. Very rarely it is now being asked, but frequently earlier it was being asked in the state board examinations. So you have to draw the double helical structure, and you can shade it to show the adenine and thymine, or guanine and cytosine bonding, the sugar phosphate backbone. So it can be given for description 2 marks for the diagram, and description you might have for 3 marks. DNA double helical structure of DNA. So nucleosome with description of nucleosome. So 2 mark diagram you have to write it, or for 2 mark question also they can ask this structure of nucleosome. This is the histone octamer, which is wound by the DNA, and there are histones that are present. So this is completely the core of histone molecules is found within this histone octamer, is found within this nucleosome model. The next one, this they have asked it with description of semi-conservative mode of replication. So you find 2 strands of DNA which has been unwound, 5' to 3' and 3' to 5'. If the strand which is synthesized towards the replication fork, and it is a continuous synthesis, the strand which is synthesized away from the replication fork is a discontinuous synthesis, which we call it as Okazaki fragments. So they are the template DNA or parental strands. So for replicating fork, they might just ask it for 2 mark questions, or they might ask it even for 5 mark questions with description, and the diagram might be for 2 marks. So the next one is this has been asked for 1 mark questions. Like the template strand is given in the question, you are asked to write the coding strand. Coding strand is usually complementary to the template strand. So now this question that they have asked is, can you now write the sequence of RNA transcribed from the above DNA? There is a template strand and coding strand. So template strand is going to synthesize the DNA. Once you have the coding strand, it is very easy to write the RNA, because the coding strand has the same sequence with the RNA transcribed, except T would be replaced by U. So it would be coding strand for RNA here, it could be UAC, G, UAC, G, UAC, G, UAC, G, UAC, G, UACG. That would be the sequence of RNA transcribed from the above DNA. So the next is schematic structure of a transcription unit. They might ask it for 2 marks. You have the promoter region, transcription starts at this site, the structural gene, template strand and terminator region. So the coding strand is here. This is what is the schematic structure of a transcription unit. This might be asked with description of transcription process or mRNA synthesis. So there are 2 strands with coding strand and template strand. So the template strand synthesizes the mRNA. It begins with the promoter region, and the process of mRNA synthesis takes place, and it terminates on reaching the end point of it. So the RNA polymerase and Rho factor are released, and mRNA is formed at the termination stage. So the process of transcription in bacteria, or it is a prokaryotic transcription in bacteria. Eukaryotic transcription is different, wherein you have the formation of G cap, and there is exons and introns that are going to happen. So all the introns are removed and exons are going to combine, and this process we call it as RNA splicing. And finally you get the mature eukaryotic mRNA with a G cap and a polyadenine tail that you can notice. So the RNA splicing, which removes the introns and retains only the exons, which has the coding sequences. So again, this can be asked for 5 marks with description, or just a diagram for 3 marks. They might ask you about the process of transcription in eukaryotes. Write a neat diagram. So the next one is the structure of tRNA, the adapter molecule. You have to remember the anticodon loop, the short arm, the other loops of it, amino acid binding site you have to remember about. So tRNA, the adapter molecule, you have this diagram part of it that you can clearly remember about. Now this is the translation process with mRNA bound by this 30S ribosome and 70S ribosome, and there are two regions, PA, that is poly, again the amylation part, polyadenine part of it, and you find the extension of amino acid that is taking place. So biological translation of protein synthesis they might ask, which involves the activation of amino acid tRNA, chain initiation, chain elongation and chain termination process. So the peptidyl binding site and acetyl binding site, PA sites, we can find in this RNA. So this process with description you are expected to draw. So the Miller's apparatus, sometimes they have asked it only for diagram width, and it should have 10 labelings. So or with description for 3 marks also they have asked this diagrammatic representation of Miller's apparatus. With description they have asked for 5 marks too. So 3 marks for the diagram and 2 marks for the description. Antigen binding site, structure of antibody molecule, they have given it for 2 marks, wherein you can notice the light chain, heavy chain and the antigen binding site with the sulfur disulfide bonds which are holding them together. So this structure of antibody molecule has been asked for 2 marks and as well as for 3 marks too. The retrovirus, this has been asked, the replication of retrovirus within the host cell. This diagram has been asked, where you have to draw out retrovirus viral RNA with RNA as the genetic material. They are going to infect the host cell, and the viral RNA is introduced into the cell. This viral RNA undergoes reverse transcription to form cDNA. This DNA gets incorporated into the host cell's DNA. Now new viral RNA is produced by the infected cell, and now they are going to produce a large number of viruses. So these new viruses which are produced can infect other cells. The infected cells can survive while viruses are being replicated and released. So replication of retrovirus, this diagram has been asked for 5 marks. This has been asked for 3 marks. The typical biogas plant, so the dung and water is fed into it, and they are going to be there, and there is an accumulation of gases of methane, carbon dioxide, and it is taken out and used for as a source of energy. This is the gas holder, and the sludge is taken up, and it is used as a fertilizer. A small amount of sludge is used as for inoculating the waste. So this has been asked for 3 marks diagram, that is PBR 322, structure of plasmid of Bolivar and Rodriguez 322, and you have to show this various restriction sites that are found on there. So you have the ampicillin resistant gene site and tetracycline resistant gene site, ORI site and ROP site, and you have to write this different restriction sites on this PBR 322. So the ROP codes for the proteins involved in the replication of the plasmid. ORI site is the origin site. So this spatched tank bioreactor. Rarely it has been asked, but it can be asked for 2-mark diagram. You can see the surface area for oxygen transfer is increased. The bubbles dramatically increase the oxygen transfer area whenever these are stirred. So that is what is the gas entrapment occurs. And here they have shown the diagram where the bubbles dramatically increase the oxygen transfer area. So the culture media is grown in such spatched tank bioreactor. The next one, this is frequently asked 2-mark questions for stirred tank bioreactor, where they have the flat bladed impeller, the culture broth, sterile air, outlet for sterile air, steam for sterilization, acid base for pH control, and a mortar. Then there is a foam breaker too. So all these labeling, it will help you fetch 3-mark question. It might be asked for stirred tank bioreactor. So this graph has been asked once or twice. The population growth curve. A represents when responses are not limiting the growth. Plot is exponential curve. So this is the exponential part of it. dn by dt equals rn. Sometimes they are just given plot the exponential curve, population growth curve. When responses are not limiting the growth, plot is logistic. That is dn by dt equals rn of k minus n divided by k, where k is the carrying capacity. This dotted line represents the carrying capacity. The y-axis has population density, x-axis has time units on it. So this diagram with description, they might also out for 2 marks, they might ask this diagram of exponential growth curve or logistic growth curve. So these are the pyramid of numbers in a grassland. This has also been asked for 3 marks. So where if you can remember the number, so only 3 top carnivores are supported in an ecosystem based on production of nearly 6 million plants. You can see only the tertiary consumer number is very less, 3. Secondary consumer is more in number. So this is a pyramid of number in a grassland ecosystem, which has been asked for many times. You have to label all these and you get 3 marks for it. Pyramid of biomass showing a decrease in biomass at higher trophic level. So you can see the primary producers having a 809 dry weight per kg per meter square, and that goes on decreasing, and the tertiary consumer has a dry weight of 1.5 kg per meter square. So that is the least that you have. So it is an upright pyramid, and you can see a decrease in biomass at higher trophic levels. So inverted pyramid of biomass of small standing crop of phytoplankton, which supports large standing crop of zooplankton. This might be given for 2 marks. And an ideal pyramid of energy, you can pyramid of energy, upright pyramid it is. So primary producers has the 10% law, you can remember about. So the sunlight is nearly 10 lakh joules of sunlight, then 10% of it is 10,000 joules. Primary consumer will have 1000 joules, 10% of that is the secondary consumer will get 100 joules of energy. Tertiary consumer gets only 10 joules of energy. An ideal pyramid of energy, we can observe that primary producers converts only 1% of the energy in the sunlight available to them in net primary into net primary productivity. So a simplified phosphorus cycle in a terrestrial ecosystem. So it begins with the rock minerals undergoing weathering and they forming the soil solution, which is uptaken by the producers, and the producers, from them the phosphorus enters into the consumers and finally into detritus, and by decomposition it enters into the soil solution. So there is 2 ways of the phosphorus entering into the soil solution. One is by weathering, another is by decomposition of the existing organisms. So this chart might be given for 3 mark or 2 mark questions. So showing species area relationship, this has been asked for 2 mark questions. So you can see on the y-axis it is species richness, and on the x-axis it is area part of it. The log to log scale, you have this log s equals log c plus z log a, and there is almost a parabolic curve, that is S equals Ca to the power of Z. Ca is Z. Next is electrostatic precipitators. So there is a scrubber, but the electrostatic precipitator has been frequently utilized. The scrubber part of the precipitator, it is clean air is there, particulate matter, dirty air enters, the water and lime or spray, it absorbs the dirt and clean air is given out. The electrostatic precipitator, which is given for 3 marks or 2 mark questions, high electric current, it is going to liberate electrons which binds to, which forms corona, and they have electrons which are released that binds to the dust particles, and they are all going to get settled at the collection plate where they are grounded, and when the clean air enters. So the biomagnification of DDT in aquatic food chain, with an example they have given, water has 0.003 parts per million. When it enters the zooplankton, it becomes 0.04 parts per million. Small fish, it is 0.5 ppm. In the large fish, it is 2 ppm, and fish eating birds will be having 5 ppm of DDT. This is an example of biomagnification of toxic substance in the aquatic food chain. So the spermatogenesis, they have asked last year, for with diagram you have to explain it. So you can, it's a very simple diagram. So spermatogonia at puberty, the mitosis differentiation takes place. There is growth of the primary spermatocytes. They undergo first meiotic division to form secondary spermatocytes. They undergo second meiotic divisions to form spermatids. So chromosomal number per cell, you can see there they were diploid 46. Here they have become 23 at secondary spermatocytes. Spermatids form, they are not mature. They undergo differentiation or spermiogenesis to form spermatozoa, which has the head and tail. So all of them are haploid from a single primary spermatocyte. So we are getting around 4 sperms, mature sperms. Oogenesis can also be asked. The chromosome number per cell is 46. Oogonia, it undergoes mitosis and differentiation to form primary oocyte. The primary oocyte undergoes first meiotic division, and they are going to undergo first polar body is formed and secondary oocyte is formed. Secondary oocyte undergoes another meiosis to form second polar body and a functional egg cell. From a single primary oocyte, we are getting a single functional egg cell. So the polar bodies, they degenerate. So it is not like 4 sperm cells being produced. Only 1 egg cell or ovum is produced at the end of oogenesis. So monohybrid cross, they have asked it for 5 mark questions many times. So you have to write the phenotype and genotype of the parents, type of gametes, F1 generation, and types of gametes by selfing of F1 or F2 equals F1 into F1. You can write this checkerboard and multiply these or cross these variety of gametes. You get the phenotypic ratio and genotypic ratio. Phenotypic ratio is 3 of them are tall and 1 is dwarf. That is 75% of them are tall and 25% are dwarf. In genotypic ratio, it is going to be based on their genetic makeup. Pure tall is 1, hybrid tall is 2, and pure dwarf is 1. The test cross that can be given to find out whether they are homozygous, recessive or heterozygous is, you can notice whenever they are homozygous, recessive, the test cross involves an unknown parent being crossed with a recessive condition, and if the results are 1:1, that is flowers are violet and flowers are white, in this example it indicates that the parent is heterozygous condition. If all of them are white, so then it indicates that the parent is going to be the homozygous parent. So 1:1 ratio, you are going to call it as you can detect the unknown parent to be heterozygous. So the dihybrid cross is also asked for 5 marks. So parent generation, you have to write the phenotype and genotype. Gamete formation, F1 generation, it would be hybrid round and yellow. selfing of F1 is F2, and gametes you can find out by multiplying R with capital R with capital Y and small r with capital Y and small y, and that is how you get RY, R small y, small r capital Y, small r small y, and you can put the checkerboard and you can cross, and you find that the phenotypic ratio is 9:3:3:1. The round is to round and yellow is 9. 3 of them are round is in green, and 3 of them are round and yellow, and 1 wrinkled green. So sex determination in honeybee, the female and male are there. Chromosome of honeybees are 32. So the females produce the ovum. If the ovum without fertilization they develop into an individual, that is parthenogenesis, they are going to be male. The ovum, if it gets fertilized by the male sperm, so they are going to be they are going to be diploid, and diploid female with 32 chromosomes. So this has already been discussed, these two diagrams. Then the switching on and switching off mechanism, lack operon concept. So switching on in presence of inducer, switching off is in absence of inducer. So you have to draw this diagram with explanation how lactose, in absence of lactose, the repressor protein binds to O gene operator region, and it prevents the structural genes, that is LAC-Z, LAC-Y and LAC-A, from synthesizing the enzymes. Whereas in the presence of inducer, that is lactose, the repressor protein gets bounded with lactose and they become inactive. So the operator region is now active, and this activates the LAC-Z, LAC-Y, LAC-A gene to undergo transcription, and they produce 3 types of enzymes: beta galactosidase, beta permease, beta transacetylase by translation process. So the life cycle of Plasmodium is very rare, but it has been given. When the mosquito bites another human, the sporocytes are introduced into the bite. The sporocytes reach the liver through the blood, and the parasite reproduces asexually in the liver cells. They burst the cell and release into the blood. The parasites reproduce asexually in red blood cells, and they burst the red blood cells and cause cycles of fever and other symptoms. The parasites, the sexual stages, gametocytes develop in red blood cells. The female and male, they are going to fuse and gametocytes are produced. The female mosquito takes up gametocytes along with the blood male, and within the mosquito host they undergo fertilization and development takes place in the mosquito stomach, and the sporozoites, that is the Plasmodium vivax, it produces sporozoites within the salivary glands of the mosquito, and when they bite a healthy individual, again the disease is transferred. The sporozoites are introduced within the by the bite of malaria, the mosquito. Mosquito is just a vector, so partial development takes place within the mosquito host. The major process takes place within the human host. So that completes our skill-based diagram-oriented questions that you can expect and you can draw. So they can even give this life cycle of Plasmodium for 5 marks. You can write it in brief. So that completes it. If you have liked this, kindly subscribe to this channel. Thank you.